Hello, Dr. Penwell and chat. Welcome back to another presentation. If you don't know who I am or haven't seen any of my previous videos, let me introduce myself. My name is Giovanna Portales, and I'm a professional presentation speaker. You may know me from my most popular video on the drug Jumperly, but if you haven't seen it yet, you can click out of this video and go watch it. For the people who stayed, today I'll be talking about ChIP assay. Now disclaimer, this won't be as entertaining as my last video, since this subject is more technical, but I will try to keep this from becoming a snooze fest. With that being said, let's begin. Now, ChIP assay stands for chromatin immunoprecipitation. It is an antibody-based technology that is used to selectively enrich specific DNA binding proteins with their DNA targets. It is critical for advancing gene expression regulation and epigenetic modification studies. The technology usually recognizes and binds histones, histone modifications, transcription factors, and cofactors. This provides information about chromatin states and gene transcription. The main point of the assay is to investigate a particular protein-DNA interaction and several protein-DNA interactions throughout the whole genomic or a subset of genes. Overall, it is a combination of proteomic analysis and molecular biology techniques used in this technology that allows for the ability to understand gene expression and regulation in cells or tissues of interest. Now, ChIP was first developed by John T. Liz and David Gilmore in 1984. The first ChIP experiments investigated using RNA polymerase and the lac operon in bacteria cross-linked with UV radiation. Later on, it was used to investigate the activity of RNA polymerase II and the topoisomerase I at heat-shocked genes in untreated or heat-shocked Drosophila melanogaster cells. However, the use of UV in ChIP was later replaced by formaldehyde, which was a more efficient and reversible method of cross-linking protein to DNA. Today, ChIP is used to study the link between histone modification and transcriptional activity. ChIP analysis originally relied on southern blots, but now endpoint PCR and qPCR are used. These techniques are useful if known genes or sequences are the subject of study. In 2001, to overcome this limitation, ChIP was developed, which is when ChIP is combined with microarray technology to provide a global view of protein-DNA interactions. Then in 2007, ChIP sequence was developed, which is a combination of ChIP and next-generation sequencing, NGS, that is mainly used today for its fast, high-resolution snapshot of protein-DNA interactions. Now, how does it work? There are 6 steps to ChIP assay. First is cross-linking, then cell lysis, chromatin preparation, shearing/digestion, immunoprecipitation, then cross-linking reversal and DNA cleanup, and lastly DNA quantifications. Sorry, that went by accident, didn't mean to click that. Now step 1, cross-linking. It begins with covalent stabilization of protein-DNA complexes. Since protein-DNA interactions are transient, in vivo cross-linking is used to covalently stabilize the complexes. In vivo cross-linking is done with formaldehyde, however it can be combined with other cross-linkers like EGS and DSG. Now formaldehyde cross-linking is great for 2 molecules that interact directly, but since formaldehyde is a zero-length cross-linking, its functionality is limited. This is why longer cross-linkers like EGS and DSG are used for higher-order interactions because they can trap larger protein complexes with complex quaternary structures. In step 2, cell lysis extracts the cross-linked protein DNA complexes from cells or tissue and brings them into solution. At this point, cellular components are free, which is done by dissolving the cell membrane with detergent-based solutions. Since protein-DNA interactions happen mainly in the nuclear compartment, it helps to remove cytosolic proteins to reduce background, which is a signal detected that is not the protein of interest binding to DNA, and also increase sensitivity. Keep in mind, detergent results will not affect the protein-DNA complex, as the covalent cross-linking in step 1 will keep the complex stable for the rest of the process. In step 3, chromatin preparation begins, which is the extraction step. That yields all nuclear material like unbound nuclear protein, full-length chromatin, and the cross-linked protein-DNA complexes. To analyze protein binding sequences, the extracted DNA must be sheared into smaller, workable pieces, which is the most difficult step to control. Now, DNA fragmentation is done with either mechanically by sonication or enzymatically by digestion with micrococcal nuclease, or MNAs. The best chromatin fragments to work with can range from 200 to less than 1,000 base pairs. Now, in sonication, it gives ideal randomized fragments, but is limited by the requirement of dedicated machinery that may need tuning, difficulty in maintaining the temperature during the sonication, extended hands-on time, and extensive optimization steps. In enzymatic digestion with MNAs, it can be reproduced easily and can process multiple samples. However, it can lead to variability due to the changes in enzyme activity and the enzyme having a higher affinity for internucleosome regions. And step 4, immunoprecipitation, where they start isolating a specific modified histone. To isolate the histone, ChIP validated antibodies are used to immunoprecipitate and isolate the target from other nuclear components. This step selectively enriches the protein DNA complex of interest and eliminates unrelated cellular material. Before starting the step, it is important to select the appropriate antibody. Now, if a qualified antibody is unavailable for target proteins, fusion proteins can be expressed in the sample, and antibodies against the affinity tags can be used to immunoprecipitate the target. Now, in step 5, cross-linking for reversal and DNA cleanup, this step is to enrich DNA bound to the protein interest. Now, DNA levels can be determined by agarose gel electrophoresis or by qPCR. Before the specific DNA products of this ChIP assay can be amplified and measured, the cross-links between protein and DNA must be reversed. This is done through extensive heat incubations or digestion of the protein component with proteinase K. In digestion, the proteinase K cleaves at the carboxy site of aliphatic, aromatic, or hydrophobic residues. Due to its broad specificity, I swear to God, apologize. Proteinase K is used to remove proteins from DNA or RNA preparations. Plus, it can eliminate nucleases from purified DNA, which prevents degradation. Then, to separate the DNA from protein fragments, phenylchloroform is used with a standard DNA purification method, or spin columns can purify nucleic acid material from complex biological samples. Lastly, in step 6, DNA quantification can be done with qPCR, PCR, microarray, or NGS. qPCR and PCR are best used for single gene analysis and can amplify and quantify specific fragments of DNA in a fast and cost-effective manner. If a microarray is used, it can create a genome-wide, high-resolution map of protein binding and protein modification. Then if NGS is used, it can align purified DNA with a previously annotated whole genomes to identify genome-wide protein binding profiles. Now let's talk about multiple ways to use ChIP assay. To do this, I will talk about 3 separate papers. In this paper, high-resolution chromatin immunoprecipitation sequencing reveals novel binding targets and prognostic role for SOX11 in mantle cell lymphoma. The mantle cell lymphoma has a specific expression of sex-determining region YBOX11 when compared with other non-Hodgkin's lymphomas. However, the function and direct binding targets of SOX11 and MCL are largely unknown. They use ChIP sequence assay to identify the direct target genes of SOX11 in a genome-wide unbiased manner and elucidate its functional significance. ChIP sequence was performed using the MCL cell line, Granta 519, as a model to identify SOX11 direct binding targets, which were then validated in MCL patients and cell lines. At the end of their experiment, they found that high SOX11 expression is associated with improved survival in a subset of MCL patients, especially in those treated with extensive chemotherapy. In application 2, in the paper, an efficient chromatin immunoprecipitation protocol for studying histone modifications in peach reproductive tissues. Rosaceae fruit trees are the best model to study the fruit growth pattern and the progression of the reproductive cycle depending on the impact of climate conditions. The Rosaceae tree research, while investigating genome structure, function, and epigenetic mechanisms, involved in important developmental and environmental response processes during fruit tree growth. Among the epigenetic mechanisms, chromatin remodeling mediated by histone modifications and other chromatin-related processes play a crucial role in gene modulation and controlling gene expression. Their goal is to figure out a plausible correlation between gene expression and the presence of specific chromatin marks. They focus on the role of 2 chromatin marks, trimethylation of histone H3 at lysine in position 4, and trimethylation of histone H3 at lysine 27. In modulating specific gene expression during the fundamental biological process in the nectarine cultivar Fantasia. In their experiment, ChIP and ChIP sequence were used to investigate chromatin dynamics in plants, mainly in different reproduction tissues of Prunus persica, to investigate histone mark distributions. As a result, they found a possible involvement of chromatin dynamics in the reproductive cycle of peach from bud to fruit, which has been observed in other species. Actually, we'll do the next one. In the last paper, Adipose tissue has emerged as an active tissue that secretes a wide range of radicals that have been related to metabolic and obesity-related disorders like diabetes, cardiovascular failure, metabolic syndrome, or cancer. However, they found that ChIP application in human adipose tissue is limited by the sample size, frozen sample availability, high lipid content, and cellular composition of the tissue. They proposed a series of improvements to the regular ChIP protocol to make this ChIP technique applicable to the study of small frozen pieces of adipose tissues. This could facilitate the study of epigenetic regulation of human adipose tissue biobanks and enhance the knowledge, as well as the role of histone in adipose tissue associated disorder. They tested ChIP for the histone marker H3K4M3, which is related to active promoters. Then confirmed the performance of the ChIP by analyzing gene promoters for factors usually studied in adipose tissue using qPCR. They found that their improvements resulted in a higher performance in chromatin shearing and DNA recovery of adipocytes from the tissue, which can be useful in ChIP, qPCR, or ChIP sequence analysis. In conclusion, ChIP assay has multiple uses that can help with researching diseases, understanding gene expression, gene regulation, and epigenetic research, and analyzing transcription factors. Thank you. And also, just as a fun fact, there were these little characters right here called Potato Chip Man has been hidden throughout the whole presentation. If you caught them, congratulations. If you haven't, well, better luck next time. Thank you.